Hello everyone, myself Prasid Unicum presenting a presentation on microscope as an analytical instrument. So let's start. We all know about microscope. Microscope is an instrument used to see objects that are too small for naked eye. Naked eye. Okay. Microscope we use daily in our laboratories, scientific laboratories for our study purposes. Hmm. Properties of microscope. A microscope, a microscope should have at least three properties. The first one is good resolution. The second one is good contrast, and the third one is good magnification. Good resolution refers to the ability to produce separate images of closely packed place objects, so that they can distinguish as two separate entities. The resolution powers of some things are given in these slides. The resolution power of human eye is about zero point 0.2 millimeter. A light microscope is about zero point 0.2 micrometer. An electron microscope is about zero point 0.5 nanometer. Immersion oil has a higher refractive index than air. Hence, use of oil enhances the resolution of a microscope. Good contrast. Contrast is improved by staining the specimen. When the stain bind to the cell, the contrast is increased. Good magnification. Accurate range with the magnification power of 10s. Magnification power of 10s means that object appears 10 times larger than its normal size. And these are the types of objective lens: scanning, low power, high power, and oil immersion. And in brackets, their magnification power is given. Total magnification of a field is product of the magnification of objective and ocular lens. Right. Scanning field have magnification power of 40 yes. Low power field has 100 s. High power field has 400 s, and high oil immersion field has 1000 s. Types of microscope. The following types of microscope are in use now: bright field or light microscope, dark field microscope, phase contrast microscope, fluorescence microscope, and electron microscope. Bright field microscope. Light microscope forms a dark image against a brighter background, hence the name bright field structure. The part of light microscope is divided into three groups: mechanical part, magnifying part, and illuminating part. Mechanical part. First one is base. It holds various parts of microscope such as the light source, the fine and coarse adjustment knobs. C-shaped arm. It holds the microscope and it connects the ocular lens to the objective lens. Mechanical stage. The arm bear a stage with stage clips to hold the slides and a stage control knob to move the slides during viewing. It has an aperture at the center that permits the light to reach the object from the bottom to the bottom. Sorry. Magnifying part. Magnifying part is consists of two lanes. First one is ocular and the second one is objective. The ocular lens. The arm contains an eyepiece that bears an ocular lens of 10 times magnification power. Microscope with two eyepieces are called as binocular microscope. Hmm. Objective lens. The arm also contains a revolving nosepiece that bears 3 to 5 objectives with lenses of different magnifying power. Illuminating part. Condenser. It is mounted beneath the stage which focuses a cone of light on the slide. Iris diaphragm. It controls the light pass through the condenser, light source. It may be a mirror or an electric bulb. Fine or coarse adjustment knob. They sharpen the image. This is the image of microscope, light field microscope, light microscope. Here we see the different parts of the microscope. Principle: the light microscope works on the principle that the rays emitted from the light source pass through the iris diaphragm and falls on the specimen. The rays passing through the specimen is gathered by the objective and a magnified image is formed. This image is further magnified by the ocular lens to produce the final magnified virtual image. This is the ray diagram of uh, ray diagram of light microscope. The light from the light source falls on the condenser lens, then on specimen. The light reflected by a specimen is captured by objective lens to produce magnified image, and then the light is passed to ocular lens to produce final magnified virtual image. Next one type of microscope is dark field microscope. In dark field microscope, the object appears bright against a dark background. It is made possible by special dark field condenser, hence the name dark field microscope. Principle: the dark field condenser has a central opaque area that blocks light from entering the object lens directly and has a peripheral annular hollow area which allows the light to pass through and focus on the specimen obliquely. Only the which is reflected by the specimen enters the objective lens, whereas the unreflected light does not enter the objective. As a result, the specimen is brightly illuminated, but the background appears dark. The application of dark field microscope is the dark field microscope is used to identify the living unstained cells and thin bacteria like spirochetes, which is cannot visualized by light microscope. Next one type of microscope is construct microscope. As per name, in phase construct microscope, the construct is enhanced. This microscope visualizes the unstained living cell by creating difference in construct between the cells and water. It converts slight difference in refractive index and the cell density into easily detectable variations in light intensity. Contrast can be enhanced by staining of the specimens, but staining kills the microbes. The property of living cell cannot be studied. The principle of dark field microscope is that condenser is similar to the dark field microscope. Um, Microscope consists of an opaque central area with a thin transparent ring, which produces a hollow cone of light. As a cone of light passes through cell, some light rays bend due to variation in density, and refractive index within the specimen are retarded by about one-fourth of wavelength. The undivided light rays strike a faceplate, a special disc located in the objective, while the divided ray miss the ring and pass through the rest of plate. The phase ring is constructed in such a way that the undivided light passing through its advance by one-fourth of wavelength out of the phase and will be cancel each other when they come together to form an image. Hmm. The background formed by undivided light is bright, and while the unstained object appears dark and well defined. The summary of application: the light ray go through the condenser and then through specimen, then through phase ring, then through objective lens, and then through accurate lens. This is the ray diagram of phase contact microscope. Application of phase contact microscope. First one is to study the unstained living cell. Second, detailed examination of internal structure in living microorganism. To study flagellum movement and motility of bacteria and protozoans. To study intestinal and other live protozoa such as amoeba and trichomonas. to examine fungi grown in culture. The, the next type of the microscope is fluorescence microscope. Refer to any microscope that uses fluorescence property to generate an image. The principle of fluorescence microscope is when fluorescent dyes are exposed to ultraviolet rays, they become excited and are said to be fluorescent. That is, they convert these invisible short wavelength rays into light of longer wavelength. That is visible light. It is red diagram of fluorescent microscope. The source of light may be a mercury lamp, which emits the ray that pass through an excitation filter. The excitation filter is so designed that it allows only short wavelength UV light, about 400 nanometer, called as exciting wavelength of light, to pass through, blocking all other wavelength rays. The exciting rays then reflected by a dichromatic mirror in such a way that they fall on the specimen, which is primarily stained with fluorescent dye. The fluorescent dye absorbs the exciting rays of short wavelength, get activated, and it turns emit fluorescent rays of higher wavelength. The barrier filter position after the objective lenses remove any remaining UV light which could damage the viewer eye, or blue and violet light which would reduce the image contrast. Application: EP fluorescence microscope. It is the simplest form of fluorescence microscopes which has the following application. First one is autofluorescence. Some microbes dilate fluorescence when placed under UV lamp. Example is cyclospora. Microbe coated with fluorescent dyes. Certain microbes fluoresce when they are stained with fluorochrome dyes. Example: actridine orange dye is used in QBC examination for the detection of Marlier parasite. Auramine phenol is used for the detection of tubercular bacilli. Immunofluorescence. It uses fluorescent dye tagged immunoglobulin to detect cell surface antigen or antibodies bound to cell surface antigen. Electron microscopy. An electron microscopy is another type of microscope. An electron microscope uses accelerated electron as a source of illumination. Because the wavelength of electron can be up to 1 lakh times shorter than that of visible light photons. The electron microscope has a much better resolving power than a light microscope. Hence, it can reveal the details of flagella, fimbriae, and intracellular structure of a cell. It was invented, electron microscope invented by German physicists Ernst and Ruska in 1932. The difference between the light microscope and electron microscope is given in the following table. Here we see the features, and here we see the abilities or parameters related to light microscope or an electron microscope. Electron microscope are of two types. First one is transmission electron microscope and the second one is scanning electron microscope. Transmission electron microscope. Electrons are generated by electron gun which travel in high speed. The medium of travel in electron microscope should be a fully vacuum part, because in air part electrons can get deflected by collisions with air molecules. Electron pathway. Electron pass through a magnetic condenser and then bombard a thin slice specimen mounted on the copper slide. The specimen scatter electron passing through it, and then the electron beam is focused by magnetic lenses to form an enlarged visible image of specimen on a fluorescent screen. Measured to increase contrast of electron microscope. Staining. In electron microscope, the stain is used are solution of heavy metal salts like lead citrate and uranyl acetate. Negative staining. The specimen is spread out in a thin film with heavy metals like phosphotungstic acid or uranyl acid. Shadowing. This technique is particularly useful in studying virus morphology, bacterial flagella, and plasmids. And the second type of electron microscope is scanning microscope. Scanning electron microscope. Scanning electron microscope has been used to examine the surface of microorganisms in a great detail. It has a resolution of 7 nanometer or less. The electron scanning electron microscope differs from transmission electron microscope in producing an image from electrons emitted by object surface rather than from transmitted electrons. Rules for using a microscope. Always carry it with two hands. Only use lens paper for cleaning of lens. Do not force knobs. Always store cover. Be careful of the card. Thank you.